Hi, my name is Shane McGlynn, and this is a medical physiology review of the digestive system, and more specifically looking at the roles of the liver and pancreas. So first we're going to start by looking at the hepatic portal system. This is just an overview to start us off. And as we can see on the diagram on the right, we have the circulatory system running through the liver. And so the significance of this is that when our nutrients are absorbed through the epithelial layers of our intestines, they're not just put straight into the circulatory system. They're actually picked up by these two capillary beds we kind of refer to as the hepatic portal system, which are then transported through the hepatic portal vein into the liver. And so the significance of this is that the liver acts as a filtration system, trying to remove any foreign particles, drugs, pathogens that the liver may deem harmful to the rest of our body and the circulatory system. So the big takeaway here is that the liver as an organ acts as an important factor of the immune system to remove any potentially harmful substances. So next we're going to look at the function of bile, which is produced in the liver. It is then stored and secreted in the gallbladder, and is secreted at the duodenum, so right at the area where the stomach meets the small intestine. And so we're going to talk about fat digestion for a little while here. As we can see, these big blue molecules are representing our triglycerides or our fat molecules. And then we have these bile salts, so obviously a component of bile. The hydrophilic side of the bile salts are on the outside, since they are water or polar, water soluble, and then fat molecules, which are not water soluble, have an attachment to the hydrophobic side. So phobic meaning scared of or not liking the water. So what these bile salts do is they'll surround this fat molecule and break it down into smaller droplets, similar to a detergent. And the reason for this is it's going to break it down to increase the surface area of the larger molecule for the enzyme lipase to act on digesting the fat. Now, something that's important to recognize here is that the bile salts actually create an outer coating or an outer boundary that the lipase cannot get through. So the pancreas actually secretes an enzyme called colipase. So what colipase does is it breaks up these bile salts so that lipase can act on these smaller fat molecules to digest them. And so when we have fat digestion, the triglycerides are broken down into two fatty acids and a monoglyceride. And so those are more absorbable forms or digestible forms of fat, since these large molecules tend to clump together, and that is not a digestible form of fat. So when these newly broken down fat particles, they are stored in micelles, so small molecules called micelles that have a different combination of different types of fats, as well as other components. So these now lipophilic fats are absorbed by simple diffusion. Cholesterol, on the other hand, is transported across the membrane of the small intestine via specific energy-dependent membrane transporters. And so monoglycerides and fat acids are then moved to the smooth endoplasmic reticulum to recombine into triglycerides. So to recap, they are broken down first, and then once they gain access into the epithelial cells, they reform into triglycerides. So from there, the triglycerides join with cholesterol and proteins to form chylomicrons, which are absorbed into the lymphatic system and transported into the venous blood. So next we're going to look at the pancreas and its role in digestion. And the pancreas is an exocrine organ that is not directly stimulated by the pituitary gland, and it produces enzymes and bicarbonate to aid in digestion. So if we look at this photograph here on the left, or this drawing, we have these acini cells that are in the pancreas, and they're really important because they're the ones that secrete digestive enzymes that empty into the duodenum. We can see the pancreatic duct meets right where the gallbladder and bile duct meet in the duodenum. So their release is very similar into their location at basically the beginning of the small intestine. And then in the pancreas, there's duct cells that secrete a watery substance of sodium bicarbonate. So most of the pancreatic enzymes actually arrive in the small intestine in an inactive form, and they need what we call trypsin upon arrival to be able to become active in their role in the digestion. So when we move on and look at the actual functions of the pancreatic juice enzymes, they have roles in breaking down a lot of macromolecules. So we can break down carbohydrates, fats, proteins, and even nucleic acids. And so the specific enzymes that are secreted from the pancreas through these Acini cells are the pancreatic amylase, which is the one that breaks down starch and glycogen into disaccharides for digestion. Lipase, which we touched on earlier, for the breakdown of triglycerides or fat molecules into fatty acids. It'll be 2 fatty acids and 1 monoglyceride. Next is protein. Sorry, this PowerPoint slide didn't come out as I wanted to, but basically it splits the bonds. These 3 enzymes split the bonds into different combinations of amino acids, proteins, and this helps the absorption and digestion of proteins. And then nuclease are the enzymes that break down the nucleic acid molecules into nucleotides. And just to reiterate, the protein enzymes are trypsin, chymotrypsin, and a carbopeptidase are what split the bonds between particular combinations of the amino acid proteins. So first we're going to look at these diagrams here on the left because we have some really good drawings, if you will, of some of the cells that we've reviewed. So we can see again we have some of the Acini cells, which are described to be in form of similar to a grape stem, where we have these little grapes or Acini cells that flow into the pancreatic duct. Down here we see the actual duct cells. So these are ones that produce and secrete the bicarbonate. And so bicarbonate production is really important, and it does require high levels of the enzyme carbonic anhydrase, which we probably remember from carbon dioxide in the respiratory system. And it acts to neutralize the acidity of chyme entering the duodenum. So the chyme that is created in the stomach is a pH of around 2, if we remember. So moving that down into the small intestine, we need to neutralize that. So this new pancreatic juice and formula through the bicarbonate at the duodenum level of the small intestine is a pH of around 9. So it tries to neutralize or make that more alkaline to protect the epithelial lining of the small intestine. So this is a very important role in not allowing the body to eat itself in a sense, and protects the epithelial lining and also creates a favorable environment for the actions of the digestive system. So that'll wrap it up. To kind of recap everything, we have the role of bile salts in the very beginning and how they are able to break down lipids. And remember that we need the secretion of the pancreas for colipase to be able to help us with the actual breakdown of the fat molecules. From there, we have really important roles of the pancreas from the many digestive enzymes of macromolecules that can break down not only fats and carbohydrates, but also proteins and nucleic acids as well. And then finally, the bicarbonate is a very important protective layer of the epithelial lining of the small intestine, as well as in the stomach, to reduce the acidity and not allow peptic ulcers to create or to become present within the stomach and the intestine. Thank you very much.